A warm welcome to the session. In this session, we are going to discuss about the accessory respiratory organs in fish. So what is the accessory respiratory organ? The accessory respiratory organ is the one which helps to breathe other than the regular respiratory organ. In fishes, normally, gills play the important role or they take the charge for the respiration. So apart from that, some other organs are going to play the major role in the respiration. So let us see what are all the organs which are going to play a major role as the accessory respiratory organ in fishes. So majority of fishes have well-developed gills for gaseous exchange, but some tropical fishes have evolved special structures, what we call as accessory air-breathing organs, to help their survival under unfavorable conditions. The following are the other organs involved in the respiration, like integumentary, air bladder, buccopharynx, alimentary canal, diverticulum of branchial cavity, and diverticulum of opercular cavity. So let us discuss about the integument or the skin or the dermal region, which is going to play a major role as the accessory respiratory organ. In several species, the skin becomes highly vascularized and will be kept moist by the mucus so that the gaseous exchange takes place between the blood and the air. These fishes move out of the water and migrate on land for a very short distance in search of another suitable water body. So at times the water body may get dried due to the season or due to some other reasons, the water may get dried. So during that time, it has to migrate from the current position to reach the suitable water body. For that purpose, the integument plays a major role. And during this migration, through damp vegetation, skin functions as an important respiratory organ like frog. So what are the organisms which has this integument of the skin or the dermal region, which is going to play as an accessory organ? Anguilla, anguilla, Amphinos coccia, Master cembulus, Rita, Periophthalmus, and Boleophthalmus. So normally these are the scientific names and common names like eel and mudskipper. Excuse me. So the next one is about the air bladder. So how this air bladder is going to act as an accessory respiratory organ. So the air bladder of some species is specifically modified for aerial respiration. So this is not for the aquatic one. This is meant for the aerial respiration, that is outside the water. So in Polypterus, Amia, Lepidosteius, Gymnarchus, and Dipnoid, all these are normally called as lungfishes. Like other organisms, like reptiles or even aves or mammals, those organisms which have the lungs for the respiration. Similar to that, they have the air bladder. So they are called as the lungfishes. And it is highly vascularized and compartmentalized into alveoli and the saculi to function as lung. So here you can see the two images which shows the air bladder. The inner layer of the air bladder has dense network of blood capillaries. So here you can see the blood capillaries like Rita mirabile, then portal veins, cilia mesenteric artery, ductus coveri, anterior cardinal vein, posterior cardinal vein, then vestigial ductus pneumaticus, which is going to make the bladder as a floating one, pneumaticus, air-filled one. So this is the image which shows how they are vascularized. And here it shows the compartmentalization. This sphincter, so this sphincter allows the posterior chamber or the oval gland to get the air filled. And from this, it is taken to the vena mirabilis or to the gas gland or to the celiac mesenteric artery. And this one is the hepatic portal vein. So inner layer, as we said, it is having a dense network of blood capillaries which facilitates the exchange of gas between the blood and the bladder. And this is about the modification of the walls of the stomach and intestine for aerial respiration. How the stomach region is getting modified. When the air bladder is there, how it is getting modified for the breathing purpose. So the wall becomes extremely thin and transparent due to great reduction of muscle layer. And the inner layer of the wall consists of a single layer of epithelial cells, which is richly supplied with blood capillaries. And it also has the mucus secreting cells and the gland cells are completely absent. Mucus secreting cells and gland cells are totally absent here because it is not going to have the integumentary respiration. So the mucus secreting cells are going to be devoid or it is completely suppressed. And in this species, air is swallowed and forced into the alimentary canal where it is stored for some time. It will be gulping the air through the pharynx, through the mouth region. It gulps the air and it is forced into the alimentary canal where it can be stored for some time. And after the gas exchange, the air is passed out through the anus or expelled by the mouth. The same way, either it will be expelled through the mouth or through the anus. And in loaches especially, the modified parts of the intestine serve for both digestive and respiration. And the digestive and the respiratory phase may alternate at short intervals, or the alimentary canal functions as air breathing organ during the summer and is non-respiratory during the winter. See the amazing fact, how the God has created this. Only during particular time, the alimentary canal is modified for the breathing purpose. That is especially during the summer region. It acts as the breathing organ. And rest of the time, that is during the winter, it acts as the non-respiratory organ and it can store the food. So only for a particular period or duration, it acts as a breathing organ. So such a wonderful modification the loaches possess. And this image shows the polypterus. And this is langnose gar, or it is also called as lepidosteus. Both these fishes are called as lung fishes. And the third type of respiration is by buccopharynx. That is, buccal region is the mouth, and through the mouth, it reaches the pharynx region. That we call as buccopharynx mode of respiration. So in some species, the epithelial lining of buccopharynx is highly vascularized, and it is having a kind of fold or pleats or tongue-like structure projecting into the buccopharyngeal cavity to form an efficient respiratory organ. So here you can see that is in Chenna. This is called as the Ophiocephalus. The common name is Chenna. And here you can see how it is having a pleat-like structure. That is, in saris, you will be taking the pleats, no? The same way, the buccopharyngeal region will be having many folds, which is used for the vascularization purpose. Example is periophthalmus, bolioophthalmus, amphipnos, Chenna, electrophorus, etc. And more advanced condition is present in Chenna species where the suprabranchial cavity. So here you can see a cavity also. Supra means always above. Branchial means gills. So above the gills, it has a cavity that normally we call as suprabranchial cavity. And this is formed in the roof of the pharynx. And these cavities are lined with highly vascularized respiratory membrane. So here you can see the respiratory membrane. So can you see this, this squared structure that normally we call as the vascularized membrane. And it has the islets. The cells are called as the islets. Here you can see the cells which normally we call as the islets, respiratory islets. And the suprabronchial cavities have alveoli and open anterior ventrally into the buccopharynx and posterior ventrally into the opercular cavity. So it opens into the opercular cavity at the posterior ventral region. All gill arcs. So it will be having the gill arcs, and all the gill arcs bear the filament, but it will be reduced in size. Due to the respiratory purpose, the gill arch filaments will be reduced in size, and on the fourth arch, filaments are greatly reduced. So this is the structure of Chenna, and here it is about the amphinos. Here also you have the respiratory islets, and this is called as the gill chamber. And inside the gill chamber, you have the air sac. And the accessory breathing organ in amphinos. Already I have shown the picture there. And it is about the bimodal breather. Normally we call this as bimodal region. See how it is laid in the land like a snake. So commonly, when a common man or a layman says, he won't say that it has a fish, because normally the fish needs a water medium for the survival. But it is of different kind, because they have the bimodal respiration. So the respiratory gas exchange takes place in a pair of specialized air sacs. So this is the air sac. So in this image, you can see clearly a white color structure, that is the air sac, extending from the pharyngeal cavity. So from here, the pharyngeal cavity will be starting and it is getting extended. Aquatic and aerial gas exchange also takes place in vestigial gills. So gills are not in a very active state. So normally we call that as vestigial, and across the buccopharyngeal surfaces and in the skin also. And this is the Chenna species, already we have discussed. And this image shows the clear structure of the suprabronchial cavity, air sac, gills, cut wall, everything. And the accessory breathing organ in Boleophthalmus. See how beautiful the organism is. See, So this is found in the swampy and the dampy places. So the opercular cavity bulges out to form two little balloon-like structure. So the opercular region will be bulging out to form, actually air sac is nothing but like a balloon-like structure. So two little balloon-like structure in the hinder region of the head. So in the head region, you will be having two balloon-like structure through the gill slits. Inhaled air, the air will be inhaled and is passed through the gill slits, stored for some time in the cavity and balloons, and after that gaseous exchange or air passed out through the bronchial aperture. And this is about the alimentary canal. Intestine. For a shorter duration, the intestine will be used as a respiratory organ, that is especially during the summer, and later on the winter duration, it will be converted to store the food. So here it is completely modified. So what are those two groups called as Loricaridae and Corbitidae? These two groups are normally called as gaps, that is gut air breathers. So when the gut segments evolve into an air breathing organ, there is generally a specialized region for the exchange of gases. So we cannot say all over the intestine or all over the stomach, the exchange of gases will be taking place, because there is a section for digestion and there is a section for breathing also. So both to be done in a similar way, but it should not be bypassed. So we have a specialized region where the gut wall has diminished, vascularization has increased, capillaries have penetrated into the luminal epithelium. So there the gut wall will be completely diminished and vascularization will be increased. That is, the blood circulation will be more and the capillaries will be penetrating into the luminal epithelium. This specialized region is generally separated from the digestive portions of the gut by sphincters. So there we have the specific sphincters, pyloric sphincters or respiratory sphincters, which helps to close the digestive sac during the respiration process. And the gut air breathing organs are generally used only for oxygen uptake. Carbon dioxide elimination seems to occur via the gills and skin in all gut air breathing fishes. See the beauty, no? So only the oxygen will be taken with the help of this air breathing organ. And the waste or the carbon dioxide is eliminated through the gills or through the skin region. So this image shows the structures. And the next one, we move on to the pharyngeal diverticulum. In Cushia eel, also gills are reduced and only the second arch bears the filament, while the third has fleshy vascular membrane. Always vascularized means it means they are used in respiration. A pair of sacs situated on the lateral sides of the head form the air breathing organ. So here you can see the vascularized oral papillae. See, folded structure that normally we call as papillae. And these sacs develop as diverticula of pharynx and line with highly vascular folded epithelium. As I said, folded epithelium normally we call as oral papillae. As it is in the region of oral, it is called as oral papillae. And the epithelium consists of vascular areas called islets, bearing rosettes or papilla. The next one is opercular cavity diverticulum. Opercular cavity diverticulum in periophthalmus and goliophthalmus. The opercular chamber serves for aerial respiration. Modification in opercular chamber. How the opercular chamber is getting modified for the respiration purpose. So the opercular chamber is large and extends below the basibranchials and above the gill arches. Basi means below the branchials. Epithelial lining of opercular chamber is thin and highly vascular. It has to be very, very thin. It should be. Not it has to be. Normally, it should be very thin. Then only the exchange of the gases and the blood can happen in a very rapid manner. And opercular bones are also thin and elastic. They have to be very elastic or in a very flexible manner. And efficient mechanisms have evolved for closing and opening of the inhalant and exhalant apertures. It has to close the inhalant and exhalant aperture. That is the opening through which the air enters. That is inhalant. And exhalant is through which it comes out. So this is the opercular region or where the gills are covered. So you can see here. And this is the mouth region. And mouth, buccal cavity, opercular cavity. Here it forms the opercular cavity and this is the gill arch. And here we have the opercular valve. See, this shows how the water enters through the buccal cavity. And the opercular cavity expands. The pressure is dropping. The pressure decreases. And the water enters the opercular cavity. The image shows how it enters in the first one and then it enters the opercular cavity. Here the water flows out of the opercular cavity. This is the exhalant siphon and this is the inhalant siphon. So it goes out and some backflow we can see always it will be there. So this picture shows how the respiration takes place. An important modification in Heteropneustes fossilis. How it happens? So it consists of two air sacs, a fan, and a respiratory membrane. See, it has a fan-like structure. So this we call as fan. And the respiratory membrane, this cells are called as respiratory membrane. And here you have the air sacs. Two air sacs are there. A pair of simple sac-like structure which is extending to the posterior, one on each side from the suprabranchial chamber of the opercular cavity, extending up to the middle of the caudal region. So you can see middle of caudal region. This from this region it is the caudal one. Until middle of the caudal region it is getting extended. And these sacs are very thin. You can see it is very thin in nature, thin walled, tubular. It looks like a tubular one and highly vascularized also. See the nature, how it is. In both the images you can see it clearly. Tubular air sac. The gut is present below the tubular air sac. See a tube-like structure. Okay. And the respiratory membrane lining of the air sac is composed of vascular and non-vascular areas and have folds and ridges to increase the surface area. Normally, when it is having a pleated structure, then it is going to increase the surface area either for absorption or for storing the air. And during respiration, fish inhales the air which enters the suprabranchial chamber through the inhalant aperture. Always it happens only through the inhalant aperture it enters, and it will be going out through the exhalant aperture or through the exhalant opening. And the modification in Clarius batrachus. Clarius batrachus. See how beautiful it is. Here also you have the arborescent organ, what we call as the rosette. Arborescent organ is called as the rosette, which would be giving a rose-like appearance. And here also they have the gill fans. The gill fans are arranged here. And the respiratory membrane, as usual, the respiratory membranes are there. And suprabranchial cavity and chamber is found. These are the modifications and how it is going to work out. So we'll see here. Suprabranchial chamber is situated above the gills by highly vascularized respiratory membrane. And two rosettes are present, one on the epibranchial of the second gill arch and another on the epibranchial of fourth gill arch. So as we said, the arborescent organs, you can see two sets of arborescent organs are there. One is on the second gill arch and another one is on the fourth gill arch. And in addition to the rosette, some gill filaments. Gill filaments are also there. So you can see here, modified gill filaments of each arch are fused and form four fans. These gill filaments will be fused and they will be forming the gill fans, one on each arc. So four gill arches will be there and each arch will be having a gill fan. So four gill arches and four gill fans will be seen. And this is a modification seen in the anabas testudius. And here also the labyrinthine organ. You can see the labyrinthine organ, beautifully located labyrinthine organ, spring-like or coiled structure. Normally we call as the labyrinthine organ. And it has the suprabranchial cavity. Here the gills are located. And this is the diagrammatic representation. And here you can see the actual picture of the anabas. So normally this is called as the Indian climbing perch. So as I have shown, the labyrinthine organ is placed on the first gill arch and it develops from the first epibranchial and consists of three concentric bony plates. It will be having, the bony plates are not represented here. So it will be having three circular or concentric bony plates. And the labyrinthine organ is covered by vascular respiratory epithelium. It will be covered by the respiratory epithelium. And this is the modification observed in the trichogaster. And here they have the suprabranchial chamber, a labyrinthine organ. You see, a labyrinthine organ is there, suprabranchial chamber is there, a labyrinthine organ is there, and gill arch 1, gill arch 2, The gill arch too will be in the form of leaf-like expression. Both the gill arches will be looking like a comb or leaf-like. Okay, it will be having a loose connective tissue covered by vascular epithelium. So you can see the structure here. And this is the labyrinthine organ. This is the modified structure. So thank you so much for this wonderful opportunity to share and discuss about the accessory respiratory organs of fishes. And hereby I acknowledge the images and the content courtesy for the Google and the various other references which has been included in this slide. Thank you so much. And in the description of this video, there is a small quiz, and the students, those who are interested, can please go and click the link to test your knowledge. A few questions, very basic and interesting questions with the pictures has been included in the Google form, and you can go and click it and you can attend the quiz. Thank you so much for the wonderful opportunity. Thank you all. We'll meet in another wonderful video again. Thank you.